this seminar series. We've got the last seminar of the academic year, and it's an exciting one, which is about causal inference methods and how they can be useful in children and young people's mental health research. We've got three excellent speakers. Dr. Francesca Solm, who is psychiatric epidemiologist and a senior research fellow and lecturer at the UCL Division of Psychiatry. And her research focuses on understanding risk factors for eating disorders in children and adolescents using large general population datasets and methods for causal inference in observational data. We also have Professor Jean-Baptiste Pingault, who is Professor of Developmental Psychopathology and Genetics at UCL, and he uses genetically informed causal inference designs, and his research aims to trace causal pathways from early risk factors to the development of mental health difficulties throughout the lifespan. And his group studies the influences of genetic and environmental early risk and protective factors on a variety of mental health outcomes. And this work draws on several disciplines, including developmental psychopathology, epidemiological psychiatry, and behavioral genetics. And we also have Eshim Jami, who is an EAS early career researcher in psychiatric genetic epidemiology. She's research fellow at the Division of Psychology and Language Sciences at UCL and is part of the iRisk project that is funded by European Research Council grant to Jean-Baptiste, and her research focuses on disentangling genetic and environmental pathways of transmission that link parental factors to psychiatric symptoms in children and adolescents using genetic data from parents and children. So I think we're in for a really exciting set of talks. I am going to let the speakers just go in the order that I've just introduced them, and without any breaks in between. So if you have questions, please put them on the chat or save them so that you can ask them in person after all the speakers have finished. And Patrizia Pezzoli, who's lecturer at the Psychology Division of Psychology and Language Sciences, will chair the question and answer session. Okay, let's get started with the first speaker.
Hi everyone, let me share my screen. And can I just check that you're seeing my full slide, not the presenter view?
Yes.
Okay, thank you. Hi everyone, it's great to be here today to present some of my research and to see some familiar names in the audience as well, and so many of you. I'll be talking about a study that we've been working on for a while, looking at longitudinal pathways between childhood BMI, body dissatisfaction, and adolescent depression using causal mediation analysis. So what I will do, I will first give a little bit of background as to why we're interested in this question and why we're interested in using causal mediation analysis to answer this question. And then I will illustrate the findings with a case study from the Millennium Cohort study and end with some conclusions. So why are we interested in this question, first of all? We know that the proportion of children that have a BMI in the overweight or obese range has increased in the past 15 to 20 years. And these are data that come from the National Child Measurement Programme about year 6 children, so children that are about 10 to 11 years old. As you can see, the prevalence of overweight has increased over the years. And at the same time, we know from large general population studies that also the prevalence of depression in adolescents has increased over the same time period. And these are data from a study that was published a couple of years ago that compared the prevalence of depression in the ALSPAC cohort to the Millennium Cohort study cohort. We also know that BMI and depression in young people co-occur, and that potentially BMI increases a person's risk of depression. And on the left-hand side here of the screen, you see the results of a meta-analysis that has looked at longitudinal studies that have looked at association between BMI and depression. As you can see, there are not many of them, but in generally, they show that there is an increased risk of depression in young people that have a BMI in the overweight or obese range. And this age group is particularly important because we know that adolescence is the time when this co-occurrence of BMI, obesity and BMI starts to become evident. And there's another study that was published a couple of years ago that showed that obesity and internalizing symptoms do not occur in childhood, but become this association starts to become manifest around early adolescence. And we can imagine that puberty and all the associated changes might play a part with it. So we also know from Mendelian randomization study, which I know you're going to hear a little bit more about in the next presentation by Jean-Baptiste. I've also shown that potentially there is a causal association between BMI and depression, although most of these studies have been done in adult population, but they have shown that fat mass is associated with increased risk of depression, and also BMI is associated with higher depression later. And studies that have looked at specific symptoms of depression have found that BMI is associated with specific symptoms like increased appetite. Other Mendelian randomization studies have also looked at the reverse association, but here the evidence has been a bit more mixed.
Francesca, I'm very sorry to interrupt. Some people are complaining about poor reception, and perhaps if you could speak maybe a little louder, or that might help, or a bit slower. That might help.
I'll try and speak loud. If I speak at this level, is it better? I'll try and raise my voice.
Thank you.
So why are we interested? Coming back to why are we interested in this question? So the goal of causality is to identify factors that are potentially modifiable, that are causally associated with specific outcomes. So the idea is that then if we modify the risk factor, then we hopefully see a change in the outcome of interest. And BMI is a modifiable risk factor. So why are we interested in specific pathways to depression? So here comes causal mediation and how we frame question and how we conduct causal mediation analysis. So the first step is to specify a well-defined causal question. So why is causal mediation needed? Why are we interested in looking at pathway and not at the exposure alone? So why are we interested in body image specifically? So what we know is that actually trying to modify BMI has been a key policy interest of the UK government for the past 20 years across different governments, whether Labour or Tory governments. And the result has been that actually these policies do not seem to have worked very, very well. As we have seen in the first slide, actually BMI in children has increased. And there is evidence when we think about BMI in relation to depression that actually the cross-sectional association between BMI and depression has become stronger in more recent years than it was in the past. So it seems like potentially something has happened in this time period. There have been some studies that have looked at pathways that involve metabolic factors, trying to explain the association between BMI and depression. But actually, although some metabolic factors appear to be involved, studies comparing either genetic loss are associated with metabolic factors with genetic losses that don't involve metabolic pathways find similar association. And similarly, when comparing people with BMI, high BMI, but a favorable metabolic profile versus an unfavorable metabolic profile, there seem to be similar association with depression. So authors have suggested that perhaps there are other mechanisms in action, and one of these to be psychological mechanisms associated with high BMI. And also biological genetic pathways are very unlikely to change in short time periods. So why body image? We know that body image has become one of the greatest concerns of young people, and the Good Childhood report that was published last year showed using data from Understanding Society that is the main concern of young people. But also our research before have shown that behaviours aimed at changing weight have become more common in young people and have become more strongly associated with depression. So one hypothesis is perhaps that the way in which we are dealing with BMI, we are trying to modify BMI in the population, or we are relating to people who have higher BMI, have caused young people to develop greater concern with their appearance and their body image, and that this might have resulted in higher levels of depression that we are observing. So the idea is that if we change this environment, potentially by changing this policy, we might be reducing some of the negative outcomes associated with higher BMI for any given BMI level, but particularly for people who have, for young people who have a BMI in the obese or overweight range. So it's not something intrinsic about BMI, but it has more to do with how we treat people who have higher BMI. So our research question would be, what would be the association between BMI and depression if children with overweight and obese BMI were not exposed to weight stigmatizing experiences, whether in policies, advertisement, and society at large, and were more satisfied with their appearance? But the issue with this question is that we can't really observe that. And that is because this is not a randomized controlled trial, but it's we have only observational data, so data about what happens in real life in the population. And this is the issue with potential outcome. We know that there are potential outcomes for every exposure, but we only observe one exposure and one outcome, but we don't know what would happen if that exposure had not occurred. And a common example to explain this concept is that if a person has a headache, they take ibuprofen and then the headache goes away, but you don't know what would have happened if they hadn't taken ibuprofen. Maybe the headache would have gone away anyway, or wouldn't have gone away. So this is the whole idea about potential outcomes and counterfactual that really is at the foundation of causal inference thinking. So because we cannot really observe, this is observational data, another way that we can frame this question is by asking how much would adolescent depression decrease if we intervene to reduce body dissatisfaction among children with a BMI in the overweight or obese range to the level that we observe in children with a BMI in the normal range. And really thinking about if we add an intervention is key to asking this type of causal question, even though in this case, actually, I don't really have yet a well-defined intervention, because we'll talk about this at the end, what that intervention might look like. So now we have a question and we have a reason for using causal mediation analysis of asking about potential causal pathway. The next step that we need to take is to choose the estimand of interest. And what do we mean by estimand? So estimate is the effect that we want to get out of our data, understand from our data. And if we think of mediation, really we have three main sort of, components of a mediation triangle, so to speak. And one is our exposure, in our case is BMI. And then we have the outcome, which is depression, denoted by Y here, and our mediator, which is body dissatisfaction. And then the important thing in this model is that these occur at different time points so that we can disentangle a temporal relationship between the variables. So the different effects that we can try and estimate from our model are the total causal effect, the natural direct effect, and the natural indirect effect in the context of our study. And what do we mean by this different type of effect? So the total causal effect is the contrast between the expected outcomes if the exposure takes the level of the exposed and the mediator takes the level that we observe in the exposed, and the contrast with the expected outcome in the outcome when the exposure takes the level of the unexposed and the mediator also takes the level observed among the unexposed. So in other simpler term, this basically is the total effect of higher BMI on depression if we leave the levels of the mediators as to how they look like in reality. Then we have the natural direct effect that really is about the contrast of the potential outcome or the expected outcome when the exposure takes the value of the exposed, and the mediator, however, takes the value that it takes among the unexposed, and contrasted with the expected outcome if we assign the exposure to the unexposed and the level of the mediator that we observed among the unexposed. So this is all a bit confusing, but in another, again, simpler term, this is the effect of the exposure that doesn't involve a pathway that goes through the mediator. And lastly, we have the natural indirect effect, which is the causal contrast between the expected outcome among the exposed when we set the level of the mediator that we observed among the exposed and compare it against the expected outcome among the exposed if we set the mediator to the level that we observed in the unexposed. And again, in other terms, this is essentially the pathway from BMI to depression that goes through the mediator. And in general, the total causal effect is the sum between the total between the natural direct effect and the natural indirect effect. So which is the estimate that we were that most relevant to our research question is the natural indirect effect. So we wanted to know if we among the exposed, if we lower the body dissatisfaction to the level that we see in the unexposed, what would be the reduction in depressive symptoms? So then the third step is to specify our causal assumptions. And in this case, our assumption an assumption of no interference of the potential outcome and potential mediator of one of the individual by the exposure of another individual. And then consistency of potential outcome and potential mediator. So we think that for one potential exposure, we would only observe one potential outcome. We would not potentially observe different outcomes for the same level of exposure. And then no unmeasured confounding of the different pathway of the mediation triangle, so to speak. So the exposure to mediator, exposure to outcome, and mediator to outcome relationships. As you can see, these are quite strong assumptions, particularly the assumption about no unmeasured confounding, which in observational data is quite a strong assumption to make, but we need to make it for the sake of conducting these analyses. So the last step would be to actually estimate the natural indirect effect and the total causal effect and interpreting our results. So I'm going to illustrate this with a study, which is not quite published yet, but you can see the study protocol on the Open Science framework with all the analysis that we have planned, including some additional analysis that I'm not going to present today. So to test this hypothesis, we have used data from the Millennium Cohort Study, which is a UK cohort study that recruited around 19,000 children that were born between 2001 and 2002, and they were residing in the UK when they were 9 months. And our sample specifically included 13,000 children, about half of them were girls and 85% were white. And so we included children who had BMI data available at age 7 when we measured our exposure, and then we imputed confounders and outcome data. So our exposure, as I said, was BMI at age 7, and we used age and sex standardized Z-score because that's how the standard way of measuring BMI in this age group. And then for our outcome, we used depressive symptoms at age 14, which were self-reported by adolescents using the Moods and Feelings questionnaire. And our mediator was a question about body image that, again, was self-reported by young people when they were 11 years old. And the question asked, how satisfied are you with your appearance? And young people could answer on a scale from 1 to 7. So before then moving on to our causal mediation analysis, we had to sort of specify what our causal assumptions are using a direct acyclic graph. And this is just a way really to explain visually what our assumption about the relationship between the variables are, so that we adjust for the correct confounders and we don't adjust for things that shouldn't be adjusted for, sort of in the simplest way possible. So we identified a number of confounders that were associated with all of these three pathways. And these were mostly either sociodemographic characteristics of the child and the family, or mental health difficulties or cognitive impairments measured prior to our exposure at age 7. But also we identify another set of confounders that were only applicable to the mediator to outcome pathway, but they were also influenced somehow by the exposure. So these were pubertal status at age 11, BMI at age 11, and mental health difficulties. Because we hypothesized that these would be sort of a consequence or influenced by our exposure, it would have been incorrect to adjust for them in the same way that we would have adjusted for all the other confounders, because that would have introduced more bias. So we need to employ methods that account for that intermediate structure of these confounders. So what we did is that we used G-formula estimation for time-varying confounding applied to mediation analysis. And there's a Stata command that was developed by Rian, Daniel, Bianca De Stavola in 2012. And then we used 1,000 bootstrap sample and 20,000 Monte Carlo simulation, and also imputed missing confounder and outcome data. And what we have done that I haven't really talked about much is that we conducted analysis on the whole sample, but also investigated whether association changed by sex, and we found evidence of interactions by sex. And so we also conducted mediation analysis stratified by participant sex. So moving on to results. First of all, we conducted linear regression to test whether there was actually an association between the exposure and the outcome, the exposure and the mediator, and the mediator and the outcome, adjusting for all the confounders that I have previously shown in the DAG. And we found evidence for all three associations. And there were also, as I said, differences. There were also differences by sex, whereby the size of this association was twice as large in girls, roughly, than in boys. And then we moved on to causal mediation analysis, and we estimated a total causal effect, which, as you can see, is very similar to the result of our main linear regression that I've shown in the previous slide. And this hopefully reminds you of that formula at the beginning that basically says what is the effect of BMI in depression if we leave, we don't account for the mediator. We think of the mediator as present among the exposed, comparing to the mediator level in the unexposed. And then we calculated a natural direct effect and natural indirect effect. And we found evidence for both pathways. So what we found was that dissatisfaction with one appearance, so our natural indirect effect, was about 30% of the total association between higher BMI and depressive symptoms. And this is after accounting for the effect that BMI age 7 had on sort of alternative pathways that could confound the mediator to outcome path, and namely pubertal status, mental health difficulties, and BMI at age 11. And as I said, all of these results, including for the natural indirect effect, were stronger in girls. I think the natural indirect effect in girls is about 40%. And we can think of why that might be the case. It could have to do with timing of puberty, and earlier onset of depression in girls, but it could also have to do with greater pressure in girls from a younger age, or a mixture of both of these factors. And why is causal mediation analysis then helpful in this context? So causal mediation analysis can be used to ask etiological questions, but as you have seen from this example, it can also be relevant to ask. It can also be used to ask policy relevant question, and it's important to frame question in terms of potential intervention. So what do our findings tell us in terms of potential intervention to improve body dissatisfaction in young people? Perhaps what our results tell us is that policies that aim to address BMI, whether in children or at population level, should consider body dissatisfaction, but perhaps even better, they should not take an individual approach and trying to change individual behavior or shame people for their behavior or their BMI, but maybe focus on a more structural approach that takes away the element of responsibility and shame and guilt in people, and that might have a stronger detrimental effect in young people, and that might play quite a large part in the association that we see between BMI and depressive symptoms in children and adolescents. So here I think I've run out of time, but just to thank everyone, Emma Blandello, who's been really working on the analysis of the study and preparing the study, and Bianca De Stavola for superb advice on causal mediation analysis, and of course all the other co-authors, and they have advice on study design and the theoretical approach to the study so far, and also the funders for this research. And thank you for listening.
Thank you, Francesca. It was a great talk. Patricia, I am supposed to go next, no? Like directly, okay. So let me just share my screen.
We'll take questions and
the
floor for discussion at the end. Yeah.
All right. Hopefully you can see that. Let me try to just move to presentation mode. All right.
Perfect.
Okay. All right, so my talk will focus also on causal inference, but using genetic data. So when trying to make causal inferences, we have a number of challenges, and today I will focus on one of them, which is genetic confounding here. During the smoking lung cancer controversy, Ronald Fisher, that is represented here, said that smoking is possibly an entirely imaginary cause of lung cancer. And his rationale was that genetic factors could directly influence both smoking and lung cancer, generating a non-causal association. So that's what is represented here, where you have genetic factors that directly impact the likelihood of smoking and then directly also impact the likelihood of lung cancer, which can generate a non-causal association. And this is basically a bit the beginning of this notion of genetic confounding. And in Fisher's word, he said that this was a common cause, in this case, the individual genotype. So this is basically the definition of confounding, which is a common cause. And here it just happens to be genetic factors. So obviously Fisher was wrong in that instance, but the notion of genetic confounding is still an important one. So what happens is that situation represented here, where you have an exposure X, you have an outcome Y, and what we're really interested in is understanding whether X causes Y. As Francesca said, the idea is that then we can target X for intervention, but then we have genetic confounding, and then we have also potentially observed and unobserved confounding. And the result is that it's basically a mess, and that's a problem in epidemiology, where you have very confounded association, and then it's very hard to basically distinguish between what is causal and what is not. So genetics is part of the cause because obviously it generates genetic confounding, but it can to some extent also be part of the solution. And we have a number of what we call genetically informed designs that can help with causal inference. So there are family-based designs like the twin design, adoption at birth design, or in vitro fertilization design, and also designs based on genomic data like Mendelian randomization or polygeny scores. So I won't have time here in this talk to go into detail. If you are interested, we just published a book with George Davis Smith and Rebecca Richmond in Bristol, basically focusing exactly on this question of how to use genetic data in order to make causal inference. What I will do in this talk instead is take a few examples of what we've done in my lab using this genetically informed design. And when I say we, I mean basically brilliant people, students and researchers in my lab rather than myself. So the first design that I wanted to discuss is the MZ twin design. So MZ are monozygotic twins or identical twin. And as Francesca mentioned, a lot of the modern causal inference literature is based on what we call the counterfactual framework. And in order to understand it, you might want to think like a bit like a mind game where you duplicate a situation and you kind of invent what we call a counterfactual situation. And it's counterfactual because it's basically impossible. So let's take the example of you give someone with depression an antidepressant, and then at the same time to the same person, you don't give that antidepressant. And so it's a counterfactual. And then if you follow that person and you can see how they fare under antidepressant or without the antidepressant, and you can make causal inference at the individual level because you can see whether the antidepressant actually worked or didn't work. But obviously that's impossible. So in real life, it often translates to a quest for the perfect match, as perfect as possible. And identical twins are matched on many observed confounders, like they have the same age, they were born at the same time, family income, etc. But they also match on a number of unknown confounders and also genetic factors. So they have by definition, they share by definition 100% of the shared environment, and then they also share their genetic material. So when there is a causal relationship, what you would expect is the twin that has been more exposed would have the worst outcome. So for example, if one of the twins is smoking quite a lot, you would expect that it increases their likelihood of getting lung cancer. And actually, many years after the controversy that I mentioned, which was in the 50s, in the 90s, there was a study that did exactly that and compared monozygotic pairs of one twin smoking and the other not, and found out the expected results of an increased likelihood to lung cancer. And on the contrary, if there is no causal relationship, because the twins are perfectly matched on genetic factors, for example, then you wouldn't see an effect whatsoever. So the first study that I want to present that we published in a few years ago focused on bullying victimization and total difficulty. So this is the strengths and difficulty questionnaires total score. So what we looked at is what we call the phenotypic association, which is basically the raw association, the unadjusted association, so a typical correlation. And what we see is the correlation is quite high, so 0.40 between bullying victimization and the total difficulty score. But then, so this is at the same time, so concurrent impact of bullying victimization. But when we control partly for genetic factors in non-identical twins, we see a decrease in that association and a further decrease when we compare monozygotic twins. But you see that the association is still, fairly strong, and is also very far from zero because these 95% intervals are very far from zero. And what we did, so from that we would say that the impact of bullying victimization on mental health is likely causal. And what we also did is follow up on the long term, and you see that the effects globally decrease. And if we focus on 5 years after the exposure, what we see is the phenotypic association is still about 0.20, but the MZ twin effect is much lower, and this time the confidence interval cross zero, and meaning that the effect was not significant anymore. And so what we see is that the impact of bullying victimization is likely causal, but on the long term it decreases, which we took as basically a sign of resilience. And what is quite important is that if you take the unadjusted association or the partially adjusted association, the effect is still quite strong. And this is probably due to residual confounding, either genetic or otherwise. And so you can see very long-term studies of the impact of an exposure like bullying victimization over several decades, and you still see an association, but it's very plausible that this association is just due to residual confounding that has not been adjusted for. So in a similar study, we look this time at the effect of birth weight and ADHD because there is a fairly consistent evidence that a lower birth weight leads to a higher likelihood of ADHD. And this time, this is the unadjusted association, which you see is fairly low, but the effect in the MZ design was this time actually higher. And that was the case at all ages, to the extent that at age 16 years, the effect of birth weight, the unadjusted association was not significant, but the MZ effect was significant. And this can happen because confounders usually, they tend to exaggerate the effect. So the correlation is actually higher than the true effect. But there can be also what we call a suppressing effect, where the effect of confounder is actually to hide the true effect. And that's what seems to happen here. And this actually has been replicated in a completely independent sample where the same pattern of results existed, meaning that the effects in MZ twins was actually larger than the unadjusted effects. So as I said, there are many other family-based designs that I won't have time to go into. But there is also more recently the use of actual genomic data in order to understand causation. And one important aspect of that is Mendelian randomization, which basically takes genetic instruments in order to get at causation. So the genetic instrument here is represented by the Z, and it is just a genetic variant or many genetic variants that are associated with one exposure. It could be like BMI, as Francesca mentioned, and then your outcome could be depression. But you're actually not interested per se in the genetic association between your variants and then your exposure. You're just interested by exploiting these, so as instruments in an instrumental fashion, to get at the causal effect here. And the reason why this is a fairly good idea is that at birth, the genetic material is randomly allocated, which means that these genetic variants are more likely to fulfill some of the assumptions of an instrumental variable analysis. So in one paper, what we did is we looked at self-harm as an outcome, and we first look at polygenic scores. So polygenic scores are basically individual scores that summarize the effect of many, many genetic variants together in one single score for individuals. And so we computed these scores for a number of potential risk factors. And what we found is that 5 of them were significant. So the polygenic score for ADHD, cannabis, depression, schizophrenia, or bipolar disorder, all predicted self-harm to some extent. So if we take, for example, depression here, MDD, we see that the prevalence in the lower quintile of the polygenic score is about 3%, and then in the highest quintile is about 4.5% of the likelihood of self-harm. So each polygenic score by itself predicts not that much actually, but when we put them together, we see that in the lowest quintile, we have a prevalence of about 2% and a prevalence in the highest quintile that is about 3 times higher, around 6%. So I wouldn't focus too much on the actual figures because there are many caveats that say that this might be slightly overestimated. However, what it shows is that the genetic liabilities to psychiatric disorders is predicting self-harm as we would have expected. So what we did, we also confirmed those analyses using actual Mendelian randomization analysis, and something that people often don't fully appreciate is that you can use also polygenic score in instrumental variable analysis, and actually under certain circumstances, the polygenic scores and the MR analysis are mathematically equivalent. But MR analysis enable you to probe a bit more the robustness of the results. And in the MR analysis, what we found is that schizophrenia, ADHD, and MDD all had a likely causal effect on the likelihood of self-harm. And what was also interesting to me is that these effects were not just everything that we tested had an effect, because we also use, for example, a Mendelian randomization analysis with genetic variants for education and a polygenic score for education, with which actually is a very powerful polygenic score, much more than ADHD, for example. And in that case, we found no effect whatsoever on self-harm. So I just mentioned how potentially to include polygenic scores. And recently we published a paper, a review paper in GCPP, focusing on the challenges associated with including polygenic scores in analysis. And so here I represent a model that is fairly similar to what I presented at the beginning, which is you have an exposure, you have your outcome, you have some genetic confounding and some confounding here from non-genetic variables. And then the difference is that you have the latent factors, so the kind of true factors, and then you have what we have measured in the squares here. This is an imperfect measure of the true factor. And this is actually quite important. And to some extent, the polygenic score can be also conceived as an imperfect measure of the genetic liability, because we know that polygenic scores usually capture a few percentages of the variance, like 3 or 4, whereas the heritability might be much higher. And so the polygenic score can be conceived as an imperfect measure of the true genetic liability. But obviously, we don't have access to these perfectly measured factors. What we have access to is the actual measured variable. And then when we fit a model with what we have, so the perfect variables, like a lot of people recently have fitted regression analysis, and they said, okay, let's regress out the polygyny score. Let's control for the polygyny score in order to basically get rid a bit of genetic confounding. But if that was the case, what we should have is that the fitted estimates, so the beta, the relationship between X and Y, should be equal to the causal estimate. But what it is actually is something like that, which is obviously very ugly. And then what that means is that the fitted estimate that you get is actually a mixture of a bunch of stuff, including residual genetic confounding, because, as I said, the polygyny score captures only a tiny bit of the heritability. And therefore, if you adjust for it, you're only adjusting for a bit of the genetic confounding. And so you have residual confounding that is left. And you also have a problem with collider bias. And you also have a problem with measurement error for all your variables. So instead of having something very clean, which is your fitted beta is equal to your causal beta, what you get is something that is actually quite hard to interpret what it represents. So recently, what we've done is that we created a method that has been published last year, which we call GSense, for genetic sensitivity analysis. And the idea here is to take the polygyny scores that we have access to, and then also external heritability estimates that we know for twin studies or genomic studies. And then we use the combination of these with the kind of intuition that the polygyny score can be conceived as the noisy measure of the latent additive heritability factor. And we kind of correct for that in a structural equation model. So this figure here represents the link between maternal education and then child education outcome. And so the first estimate here is kind of the unadjusted association, and it's 0.40, which is quite large because we know that maternal education is actually the strongest risk factor for child educational attainment. And then what we did here is we just adjusted for polygyny scores that we had computed. And as expected, when you adjust for the polygyny score, the effect decreases because in a way you adjusting for some of the genetic confounding. But if you adjust for the best fitting polygyny score, which even for education is captures quite a lot of the variance, you still have a quite substantial effect here, but you're not, again, adjusting for all of the genetic confounding. So when we conduct the sensitivity analysis, we say, okay, what would happen if we had a polygyny score that captured all the genetic variants, and so captured more and more of the heritability here. And so as you go along on this x-axis, this corresponds to higher levels of heritability. And recently there was, so if you go to the extreme where you have a heritability of education of 0.60, which is actually the estimate in many studies, you have a kind of effect of maternal education on child outcome, which is very small. And then recently there was a meta-analysis which estimated heritability for education of around 0.40. And what you see here is that the effect of maternal education on child achievement decreases greatly, but not completely, meaning that a large part of this effect is likely due to genetic confounding, and there is potentially a residual effect that is not due to genetic confounding. So I don't have much time to talk about the many other approaches combining genetics and causal inference, but I wanted to talk about a paper that is currently in revision. And in this paper, what Tabia did was to basically fit a common liability to a number of substance use issues like cannabis, cigarette, and alcohol. And what that enables us is to have more power to try to understand what is shared between those different indicators of substance use. And doing that, she was able to find 42 genetic variants that were significant, and 60% of them were novel in the sense that they had not been detected for any of the indicators before. And what is actually quite interesting is that some of these variants that are around here, for example, here you have DRD2, which is dopaminergic genes. And these genes have been suspected for a long time to be involved in the common liability, but in in studies that look at single genes, or even in previous GWAS, they've not been identified as genes underlying the common liability. And so by using this technique, we are able to have a much better understanding of what are the common risk factors that explain the co-occurrence between different types, different classes of substance use. And this can be used in downstream analysis, not only to better understand what creates a common liability to substance use, but also to potentially interrogate the causal relationship between this common liability and other risk factors, etc. So this is part of a set of new techniques, which, including MR techniques, which are discussed in more detail in the book, that enable researchers to better understand the kind of molecular pathways from genetic influences to outcome, and even potentially to identify new targets for drug development. And all of these developments are based on these methods like MR, which enables better causal inference by using genetic data. So thank you for your attention. And again, I can't acknowledge everyone who contributed to these studies, but they are great researchers. And thank you to the founders. And I will leave Eshim, who is one of those great researchers in my lab, to present talk. So,
thanks, Jerry and Francesca, for really interesting talks. I'm just going to share my screen. Can I get some verbal confirmation that
yes, it's perfect. Thank you.
Okay, great. So I'm going to also talk to you about genetically informed designs, but this is in the context of research which looks at the transmission of mental health behaviours within families. And I'll also share with you the results of an ongoing study on this topic, which I'm working on. There you go. Okay, so it's commonly observed that psychiatric disorders run in families. This table is from a large study by the World Health Organization, including 55,000 individuals from 22 countries, in which they looked at associations between parent and offspring psychiatric disorders. And what they found, which is shown here in the odds ratios, was that individuals with mood, anxiety, substance use, and behavioral disorders were 2 to 3 times more likely on average to have parents who also had a psychiatric diagnosis. And this correlation was present for both similar and dissimilar parent and offspring psychiatric diagnoses. So, for instance, between the parent's depression and the child's mood disorder, but also between the parent's depression and the child's substance use or behavioral disorder. There are other parental characteristics which have also been linked to the children's mental health behaviours in the literature, and this can include factors related to the parent's personality, parenting behaviours, and also their educational attainment, which is the number of years spent in education. However, when we look at any association between a parent phenotype and a child outcome, we can't necessarily say that these parent behaviours or characteristics have a causal effect on children's mental health outcomes. That's because an important confounder, which may be present in any association between parents and children, is the role of genes. So as each parent passes on half of their genes to their child, these genes can have a direct impact on the child's outcome through this genetic pathway. This makes it possible that any observed parent and child association could be explained by this genetic transmission pathway, rather than this direct impact of the parent's phenotype on the child's outcome. So parent-child associations can also be confounded by gene-environment correlation. And passive gene-environment correlation is when parents both pass on their genes and provide an environment to the child which could be related to their outcomes. So, for instance, if we take the example of depression, then the association between parental depression and the child's outcome could be explained by both the depression-associated genes inherited by the child and the depressive symptoms of the parent that the child is exposed to during development. Evocative gene-environment correlation is another type where the child's genetically influenced characteristics evoke a response from their parent. So in this case, an association between a child's outcome, such as depression, and a parent's behavior, such as harsh parenting, could actually be explained by an evocative effect in which the child's genetically influenced behavior, which could be something like not tidying up their room or not finishing off their food, that's evoking a harsh response from their parent. And the association isn't due to a causal effect from parent to child. So to tease apart these different pathways of transmission, we need to use genetically informed intergenerational designs. And recent methods use genetic data from families to do this. And one example of this is the trio design, which uses information from mothers, fathers, and children. So these designs capitalize on the fact that genetic variation can influence an individual's outcome indirectly through the environment, which is under the influence of heritable traits. So in this diagram, the purple pathway going from the genotype of the mother and the father to the child shows genes passed on to the child, and the effect of these genes on the child's behavior, which in this example is ADHD, is a direct genetic effect. However, these green pathways, M and F, from parent's genotype to the child's ADHD symptoms, show that the parent's genotype can also influence the child indirectly through the environment, through genetically influenced parental traits. And this is described as an indirect genetic effect. So the way that we estimate this effect is to control for the genotype of the other parent and the child within the predictive model. And this gives us an estimate of maternal or paternal indirect genetic effect that is mediated through the environment. Okay, so this brings us to the ongoing study, which is about familial effects on early environmental exposures during childhood that can put children at risk of experiencing mental health difficulties. So we're lucky enough to have access to the Norwegian Mother, Father and Child cohort study called MOBA, which is a nationwide population-based cohort in Norway. They have extensive data on parents and children, and it also uses a longitudinal design with long-term follow-up, which gives us the opportunity to look at parent and child behavior throughout the developmental trajectory of the child. It's also one of the largest pregnancy cohorts in the world. So this rich data set has allowed us to extend previous work by not only looking at effects from parents to children, but looking at how all trio members influence each other during the child's early developmental period. So in terms of early environmental exposures, we focused on several different variables that capture this early risk. And that includes parents' psychiatric symptoms, which we looked at ADHD and anxiety and depression. It also includes parents' life satisfaction, relationship satisfaction, their parenting behaviors, whether the child has experienced parental divorce or separation, and also whether the child has been exposed to bullying. And these are all captured through questionnaires that were administered to mothers and fathers. For familial effects, we wanted to focus on the influence of parents and children's psychiatric traits, including ADHD, anxiety, depression, and schizophrenia. And we also included educational attainment, as there's been some indication of indirect genetic effects of educational attainment in the literature. So these traits were indexed by calculating polygenic scores for each trio member, which means that we didn't have to directly measure these traits in the sample. So we used regression models to look at whether polygenic scores for these traits for each trio member were associated with the variables that captured early environmental exposures. And what we were really interested in is whether in understanding what type of an effect explained an association. So we did this by looking at whether maternal, paternal, and children's polygenic scores for these psychiatric traits and EA were independently associated with these early environmental exposures. And then we used multivariate models to see whether the association between these polygenic scores and the environmental exposure was still present after we accounted for the polygenic score of the other two trio members. So through our models, we were able to detect whether 4 types of effects were present on these studied early environmental exposures. So for each of these effects, it depends on whether polygenic associations remain after accounting for the genetic effects of the other trio members. So firstly is what I also showed earlier, which is an indirect genetic effect from parent to child. And we see this association between a parent's polygenic score and a child's outcome. This would indicate a parent to child indirect genetic effect. If we find an association between the child's polygenic score and the parent's outcome, this suggests a child to parent effect, which indicates evocative gene-environment correlation. If a parent's polygenic score is associated with the outcome of their partner, this indicates a partner-based indirect genetic effect, where one parent's genetically influenced trait is influencing their partner's behavior. So all of these are indirect genetic effects, but then finally, we can also look at passive gene-environment correlation. And this is indicated if the child's polygenic score is no longer associated with the parental exposure, meaning we don't find evidence of an evocative effect. So this would suggest that the earlier parent-child association, which we found, is actually explained by passive gene-environment correlation, in which the child has inherited the parent's genes, which are also related to the parental exposure. Okay, so we found that many associations between psychiatric traits and early environmental exposures were explained by passive gene-environment correlation. So for instance, children's ADHD polygenic scores were associated with parental divorce or separation initially. But after we accounted for the parents' polygenic scores, this association disappeared, which indicates that there was no evocative effect of a child's ADHD on the parents' divorce. This suggests that the earlier association is best explained by a passive gene-environment correlation, in which the child's inherited risk of ADHD is correlated with the parental divorce that the child is exposed to. And then, as shown here, we observed similar associations for the other psychiatric traits. So depression, for instance, was also linked to maternal anxiety and depression, and then also to maternal life satisfaction through passive gene-environment correlation rather than an environmental effect. So in terms of indirect genetic effects in parent-child associations, we found no strong evidence of that in this study. But something to bear in mind is that it doesn't necessarily mean that environmental associations don't exist between parents and children for these variables, because the trio design, as JB also discussed, actually the use of polygenic scores has limitations, and the trio design also requires a lot of power. So what we can do to better understand whether environmental effects are truly absent or present is to compare whether evidence from multiple designs is consistent with each other. Interestingly, we did find some evidence of indirect genetic effects in partner associations where one parent's genetically influenced trait was associated with the trait of their partner. So, for example, both maternal and paternal schizophrenia polygenic scores were associated with their partner's relationship satisfaction. And then similarly, the educational attainment polygenic scores of both the parents was negatively associated with whether they were divorced or separated. These findings give us interesting insight into effects not just between parents and children, but between the parents themselves during the child's early developmental period. So to summarize, in this talk, I discussed how associations between parents and children are prone to genetic confounding. And this makes it really important for us to use genetically informed designs to disentangle genetic and environmental effects. The availability of genetic data in family-based cohorts such as MOBA gives us exciting new opportunities to understand pathways of transmission within families. But ultimately, this knowledge could help us to refine treatments and prevention strategies, for instance, by being able to identify individuals at risk early on, or seeing whether interventions in one generation can impact the other, or also whether interventions should target the parent generation or both the parent and child generation. That's all from me, and thanks to all my colleagues at UCL and MOBA who collaborated with me in this work.
Thank you. Thanks to all the speakers. Thanks for the extremely clear and extremely informative talks. I think they really speak to how much the field has advanced in terms of kind of from traditional cross-sectional designs that didn't really enable us to account for causation, and also speaks to kind of the potential that we have to exploit existing data sets using the new advances, the new analytical approaches. So thank you, this was brilliant. There are currently no questions in the chat, so what, Essie?
I have a question generally for JB and Eshim. So when you use, I mean, it's very nice and neat thing to do to use this polygenic risk scores and look at the prediction. But I have always sort of wondered to what extent we have to sort of tread a bit carefully in that obviously these things are only as good and informative as the polygenic risk scores are. And also presumably polygenic risk scores are formed based on the kind of variants that sort of are common enough and have big enough impact in the population that they get picked up. So I'm wondering to what extent, so this is a slightly different question from the first part, to what extent do you think these sorts of polygenic risk scores are helpful at the individual level? So to what extent are they helpful if you think about translational impact at the level of an individual? Because by definition, you're picking up the things that are common in a population. And of course, if an individual has those variants, then they're probably quite helpful. But you may have some genetic variants that are incredibly important, but because there may be rare variants that they don't get picked up in GWAS or something, there might be some individuals where you can't currently model these sort of genetic confounds very well, and you can't also do individual prediction very well.
Yeah, I mean, as you said, like the polygenic score capture often a tiny bit of the variance. That said, it's not always a massive issue, and the reason is like when you look at MR analysis, even if you have a small instrument, so an instrument that predicts enough variance in the exposure, but it's often tiny amount of variance. But what is really important is that the instrument effect is unconfounded. So in a way, it's better to have a small effect that is unconfounding than the massive effect that basically will be confounding as a correlation. So for example, in MR, obviously the individual thing like instruments we use account for a tiny bit, but if you have good instruments, even if they account for a tiny bit of variance, you are able to retrieve causal effect like we think appropriately. That said, for the polygenic score, there is a question of power, and that's something that Eshim mentioned. That's why we can't say for sure, as we can never really say for sure that there is no effect, because we would have to make sure that we have massive power. And we did a few calculations for these trio designs some time ago, and we would need something like 20 to 40,000 trios, depending on how big the effect is. But these are figures that we're getting at now, like the new release of MOBA has more than 40,000 trios. Another thing, and then Eshim might be better able to talk about that, is that sometimes we don't use, there is new methods that don't use the actual polygyny score, but use methods like SNP heritability that use all the variants. And then Eshim has just published a paper using one of these methods to look at the trio design. Is that correct, Eshim?
Are you referring to GCTA?
Yeah.
Yeah, so I have a paper on that from a couple of years ago, and there's another one which is submitted. Yeah,
so basically these papers, they model all the common genetic variants, and therefore you have potentially more power. But they are, like as Eshim said, also you kind of need to look at basically evidence from all many design, and not just polygyny scores, but all kinds of design. To respond to your second question, which is slightly different, is how polygyny score will be useful clinically. I think the answer is still up in the air, basically. So some work has shown that for cardiovascular disease, adding the polygyny score will add to the prediction at the individual level, and you would be surprised how the questionnaires that they use to assess your risk for cardiovascular bad cardiovascular outcome is actually quite poor in terms of specificity and sensitivity. And then the polygyny score adds a bit, but anything is good in that sense, and that might be closer to kind of clinical utility. And more there is evidence that they will be able to be used and be clinically useful. Whether that will also be the case for psychiatric disease is another matter, and I think the matter is not completely settled yet.
Thank you very much. We have one other question from SE for Francesca, which is, where would you like to go next with your work to maximize translational impact?
Yeah, good question. So I've got some ideas that are in the process of being evaluated in the form of a fellowship application to really address this issue. I think one of the issues whether indeed body dissatisfaction is causally associated with depression, and that is one of the things that I mentioned in my presentation, that a lot of the assumption in that study relied on the idea that there was no unmeasured confounding, and as we've heard from the other two presentation, we can never exclude genetic confoundings in this association. So one bit would be to really establish that causal association, and some other ideas I've had are to use some natural experiments, which are other designs for causal inference that we haven't quite discussed today, but things like regression discontinuity to evaluate the effect of existing policies and whether those actually do increase the risk of mental health difficulties and potentially via increased body dissatisfaction. So those would have very straightforward policy and translational implication, as well as better understanding than what causes body dissatisfaction in young people. So yeah, that's kind of what I am trying to do next, to really take forward this program of work. So fingers crossed I get the funding.
We will keep our fingers and toes crossed. Absolutely. Thank you. And we have another question from John. For anyone who wants to answer, it might be Jean-Baptiste and Eshim, perhaps. So recently there's been a lot of discussion about the extent to which polygenic risk scores derived from unrelated population samples may themselves be confounded, for example, by stratification. And as John understands it, this can be addressed using family-based polygenic score analysis, which typically produce smaller estimates, suggesting that these confounds are real. So should Mendelian randomization analysis be using these family-based polygenic scores instead?
The short answer is yes. So John, your understanding is perfectly good here. The polygyny score can be confounded by things like population stratification, and then within family analysis can answer that. And actually, a paper was recently published in Nature Genetics, looking at GWAS, so genome-wide association studies, but within siblings, and this would take care of the population stratification. And they did follow-up analysis, and I'm involved also in another paper looking at that for the impact of education on mortality, for example, looking at whether follow-up analysis using summary statistics derived from this corrected, within family corrected summary statistics would produce different estimates. And the answer is a bit more surprising than we would expect, is that yes, there is the deflation. So the kind of variance explain less within family because they corrected for all these problems, including population stratification. But this doesn't necessarily affect too much the MR estimate, because often the association with the exposure, but also the association with the outcome are affected in the same way. And so the kind of ratio, the causal effect, is not affected that much. But the short answer is yes, like all this kind of normalization analysis would be better off using those kind of within family designs, in particular for phenotypes where there is a bit of inflation, like education, which is not something we observe for other phenotypes that are more medical, like LDL cholesterol, or all these kind of things are not really affected as much by issues like population stratification and genetic nurture.
Thank you very much. I would like to close on time because I know that there is another event starting shortly. And I want to thank the speakers for the illuminating talks and everyone who asked questions and contributed. And I also want to remind you that we will have an in-person early career researchers event in children and young people's mental health that will showcase early career researchers' work at UCL. So I've just popped in the link to register in the chat. So I really hope to meet you there. Thanks so much again for your time and contribution. And if there are no further questions, I guess we can end. We can end now.
Thank you.
Thank you.
Thank you, everyone, for your time. Bye.
Bye.
Bye.